Okay, so from glycolysis, we just made 2 NADH, 2 ATP, 2 pyruvates. Then we go through pyruvate dehydrogenase complex, okay, that transforms pyruvate into acetyl-CoA using vitamin B1, B2, B3. So we're using thiamine. We're using a lipid that has sulfur groups. And then we also have an FADH needed, and we produce NADH. And then we, of course, we need a decarboxylation reaction as well. So we're producing carbon dioxide. Finally, we get acetyl-CoA. We mix that with oxaloacetate. Oxaloacetate is an alpha-keto dicarboxylic acid, and it has 4 carbons. Then we go from acetyl-CoA into citrate, and then we go from citrate to isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and back to oxaloacetate. We're making FADH, GTP, NADH, and CO2. The rate-limiting step for this citric acid cycle is the isocitrate dehydrogenase complex. Note that the alpha-ketoglutarate dehydrogenase complex is very similar to the pyruvate dehydrogenase complex because it produces carbon dioxide. It also adds a CoA, and then we're getting succinyl-CoA. And then notice that GTP is the same thing as ATP almost. Okay, so that is that. Alright, so let's go through fatty acid synthesis phase. So citrate kind of builds up because oxaloacetate is already used for gluconeogenesis, so the citrate is building up. And that citrate goes out into the cytoplasm from the mitochondria, and it breaks down, we get a citrate lyase that breaks it down into oxaloacetate and acetyl-CoA. Oxaloacetate is going to be transformed back into succinyl-CoA to go back into the mitochondria, and at the same time we're producing NADPH. NADPH, as I mentioned in the previous video, is important for reductive biosynthesis. In this case, it's going to be for the synthesis of fatty acids. Acetyl-CoA is going to be joined to malonyl-CoA using an acyl carrier protein. We're also going to use a series of reactions to produce NADPH, to produce fatty acids. And we're requiring, of course, NADPH. Okay, the only fatty acid that we can make is a 16-carbon fatty acid called palmitic acid. Okay, to do the opposite of fatty acid synthesis, of course, is a reduction reaction that creates a fatty acid. The opposite is an oxidation reaction. That would be the breakdown of the fatty acid using beta oxidation. So what does that mean? We're oxidizing the beta carbon. Okay, so we oxidize the beta carbon, we get an OH in there, then we oxidize the OH again into the ketone, carbonyl, and stuff like that, and we keep on going. And we generate NADH and FADH. And from that, after we get acetyl-CoA. Acetyl-CoA can actually join another acetyl-CoA molecule and form acetyl-acetyl-CoA. And then we can get HMG-CoA. And then from HMG-CoA, we can do 2 things. We can either transform it into cholesterol by using HMG-CoA reductase. We have to go through mevalonate. And eventually we get a series of reactions that are adding terpenes and isoprene units, and eventually we get cholesterol. Or we can use HMG-CoA directly to make ketone bodies. So ketone bodies are similar to acetyl-CoA, okay? And we just use those for when we're starving. So when we're running out of energy, we don't have glucose. The only thing that we have left is our ketone bodies, okay? They generate more energy. So from ketone bodies, we can just we go from ketones to acetyl-CoA, okay? To get that energy. Acetyl-CoA provides the energy. We cannot go back to glucose from ketone bodies. Alright. So the next thing we have to take into consideration are the shuttles. We have the glycerol phosphate shuttle, and then we have the malate shuttle. So in glycolysis reaction, we made 2 net NADH that are in the cytoplasm, but how in the world do we get them into the mitochondria? We're going to use these shuttles for that. So the first one is glycerol phosphate shuttle, and eventually it's going to become, it's going to yield from NADH to FADH. So it's producing FADH from NADH. We have a total net loss of 1 ATP. FADH it produces 1.5 to 2 ATP, okay? So around 2 ATP are produced, whereas NADH produces 2.5 or 3 ATP. Okay, so the malate shuttle is more effective because you go from oxaloacetate to malate to oxaloacetate, okay? So malate is a good molecule because it's like a car. It goes from the cytoplasm into the mitochondria, so it can easily be transported in and out. And that's where we go from oxaloacetate in the cytoplasm to malate in the cytoplasm. Malate can go through the mitochondrial membrane. You know, the outer membrane is very permeable to big molecules, but the inner membrane is not. But malate is permeable, okay? So the inner membrane is permeable to malate. I guess there's a shuttle. And the malate eventually becomes oxaloacetate back in the mitochondria, generating NADH. And of course, we know this reaction in the citric acid cycle when we go from malate to oxaloacetate, making NADH. Right? So that is easy. Those are the shuttles. Now let me talk about the amino acids. You know, there's ketogenic, and there's gluconeogenic, and there's both in terms of amino acids. I have a mnemonic device that I came up with because of my professor. It's called Very Frequently I Work Like Mr. T.K. Harris. Mr. T.K. Harris was the name of the professor. So I just have that abbreviation for all of the essential amino acids. And what I did was I basically said that for the win, Y and I represent amino acids that can be both, okay? Gluconeogenic and ketogenic. And the only ones that are ketogenic I labeled here that are lysine and leucine. So those are the only ketogenic. The rest that you see here are gluconeogenic, okay? So the only one that are both are FTW, so for the win, and Y and I. Hopefully you know the abbreviations for the amino acids. If you don't, please learn them. Okay. So that will be it. Also in terms of how you convert one amino acid into something else, how do you convert oxaloacetate into asparagine, into aspartate or asparagine, and glutamate into alpha-ketoglutamate? Well, we use what are called aminotransferase. So a aminotransfer is going to transfer an amino from, for example, glutamate. Glutamate has an amino group. So we transfer the amino group, and then we transform glutamate into alpha-ketoglutamate. And at the same time, we're attaching it to oxaloacetate. So the amino with the oxaloacetate is going to produce an asparagine. So they're both 4 carbons, and you can tell that they're similar. Okay, so we went through the shuttles. Okay, so if we want to go through gluconeogenesis, we have to go back, and there are some irreversible steps in glycolysis. We have to overcome those. So the first thing that we have to do is to transform pyruvate into oxaloacetate using a carboxykinase. A carboxykinase generally adds carbon dioxide, and it's B7 dependent, so it's biotin dependent. Anything that adds carbon dioxide probably depends on biotin. Anyway, so you get oxaloacetate, and then from oxaloacetate you go into malate using the malate dehydrogenase enzyme. And then from alate, which can easily go out of the mitochondrial membrane, okay, and then we use a phosphoenol pyruvate carboxykinase. Carboxykinase is going to add a phosphate kinase, and a carboxy, it's going to add a carbon dioxide, okay? And then we get phosphoenol pyruvate, okay? So we get phosphoenol pyruvate, and then we go all the way up. We got fructose 1,6 biphosphate. We gotta bypass the PEP CK enzyme. So how do we do that? It's gonna be fructose 1,6 biphosphatase. It's gonna remove the phosphate from fructose 1,6 to convert it into fructose 6-phosphate. And eventually we go through glucose 6-phosphatase, and then to go back to glucose. And then if we want to go through glycogenesis, making glycogen, one thing I have to mention is that amylopectin is alpha-1,4 and beta-1,4, and it's alpha-1,4 and beta-1,4 branching, okay? So we need to have a branching enzyme in order to make it. Okay, so the first thing that happens is we get from glucose, glucose is attached to UDP to get UDP glucose, and then this is called the glycogen synthase enzyme that eventually forms glycogen out of that. And then to go backwards from glycogen into glucose 1-phosphate or into glucose, we need to have glycogen phosphorylase, okay? It's a phosphorylase. It's going to break away those glycogens. The last glucose that comes out does not have a 1-phosphate in it. It's going to be just free glucose, but that's the last one. All of the others have glucose 1-phosphate. Okay. So we went through pretty much everything. Let me see if I'm missing anything. Okay. That is pretty much it.